First of all, I'd like to thank Hugo for accommodating to my need to go to my granddaughter's birthday party this afternoon. So I don't know why this is, whether it's showing to everyone, but it seems to be recording me. So anyway, I'm looking really at the difference between the function of satellite cells during growth and during regeneration, and to see whether the differences between them are in any way an impediment. Remember that Duchenne boys are growing for the first 18 years of their life, or trying to, and at the same time they're having to repair their muscles. And you need to know whether those two functions are in any way interfering with one another. So the mouse both grows and regenerates. So this is a normal mouse muscle, and this is a MDX mouse muscle, and it is degenerating and regenerating. So this is an area of degeneration, this regeneration, in response to the lack of dystrophin in the muscle fibers. The cell responsible for this, as well known, is the satellite cell. And the satellite cell has been highly characterized in terms of the molecular biological events that occur during this process of regeneration. So there are a series of stages that the satellite cells go through, each of them marked by a pattern of transcription factors, ending up in a cell that becomes a muscle fiber, or enters into a muscle fiber, or repairs a muscle fiber. And there is some debate as to the extent to which each of these can go back to be, stages can go back to dormancy. If we look at what happens during the growth of the MDX mouse, these are muscle fibers taken from a series of wild type mice on this side up to 6 months, and MDX mice, and you can see they both grow. The MDX mice do seem to be a bit deficient when they're young, and I won't go into that for the moment because it's a complicated story. But one of the things you must do if you're trying to analyze growth and regeneration is to get reliable measurement from it. So learning from our physicist betters, we should do the best we can to get reliable, good measurements. So Bill Duddy, some years ago in the lab, started to take out muscle fibers and stained them with fluorescent phalloidin, which marks all of the fibrillar actin. He could then measure the total volume of each fiber. He could also count the number of nuclei present in each fiber. And so you have two bits of data related to growth, one from the satellite cell point of view and one from the myonuclear point of view, which describe growth. So this is the evidence that the fluorescent phalloidin staining gives you a good measure of size. So this is the measurement, the phalloidin fluorescent yield per fiber of for a number of fibers, both MDX in red and wild type. And this is plotted against the nuclear domain, basically, as measured by confocal stacks. And you can see that up at the top end here, these muscle fibers, these normal muscle fibers especially, have a nuclear domain in the 20 to 30,000 cubic microns rate region, which is as big a nuclear domain as you see in any tissue. The fibers of the MDX mice, as they get older, begin to branch extensively. And so we measure the yield from the whole muscle, including all the branches. So we have a true measure of the volume of those muscle fibers and of the content of myonuclei. I like to display my data in a rather peculiar way that no one else seems to like, but I find it descriptively useful, pictorially useful. So this is a horizontal scattergram of an MDX mouse muscle and a wild type mouse muscle in terms of the number of nuclei present in them. What I like to do is to stretch those data over the rank. So I rank them, and so this is the highest ranked fiber. This one contains the most nuclei, and these ones down here contain the least nuclei. And it shows you a roughly Gaussian distribution, with the exception of this little group down here, of the numbers of nuclei per fiber. And that's useful because you can then follow the transition in terms of whole populations. So for instance, this is that same plot with a longer x-axis, and that's week 1, and this is week 2. And you can see that both the MDXs and the wild types have moved the same distance, that both populations have basically shifted to the right. And the same happens if you look at 3 weeks, but by 3 weeks it's beginning to slow down, and by 4 weeks it looks as if it's almost stopped. So Pete Zammit has very similar data to this from his lab, quite independently, and almost all our numbers correspond to one another. What's important here is that we know how many nuclei have gone into each fiber over that 1 week period from satellite cells. So we can begin to do calculations. We know that there are 90 myonuclei per week going into a fiber. That's 12 to 13 per day. And that's accomplished. So that's 1 myonucleus every 2 hours is going into these fibers to get them from there to there. So how many satellite cells are responsible for this? Well, there's between 5 and 8, I think. It was also a piece of data that Pete and I agree on. Pete's lab, my lab agrees on. And so you can then calculate how much work they have to do. So basically, in order to get from there to there, you need to without increasing the number of satellite cells, because they are either remaining stable or slowly declining in number at this age, you need a serial asymmetric division of each cell. So each satellite cell has to divide asymmetrically about twice a day in order to accomplish that growth. So we know that this is a asymmetric division. You can't do this with serial any significant amount of, sorry, this is mostly asymmetric division. You can't do it with, you can't include a decent amount of symmetric expansion in there. So that's a picture of what's going on during the very early growth period in these mice. At this time, we thought, and Pete Zammit also thought, that everything stopped at 3 weeks in terms of putting more myonuclei into muscle fibers. But when we looked more carefully, we found this wasn't so. So this is a 4-week wild type mouse. This is a 5-week wild type mouse. And this is an 8-week, sorry. And this is a 14-week. So you can see that between 4 and 14 weeks, there's also been an increase in number, and it's also about 90. But this 90 nuclei that have got into each of those fibers during that period, which is just growth, these are wild type animals, is about 1/10 the rate that was happening during week 2. So it's carrying on in a similar way, but at a much, much slower rate. We've not really penetrated what's going on there, because it's a very subtle, discrete process. We only pick it up by these cumulative measures. So this is the same graph, again, condensed along the x-axis to allow for a bit more. I don't know what that's doing there. So this is what male mice do. So all these mice here are female mice. Male mice grow slightly bigger over this period. And these lines here of the blue symbols are what MDX mice do over this period. So you can see that MDX mice grow hugely in terms of the number of myonuclei per fiber. They are sort of 2 to 3 times the number of nuclei, if you go along the median here, for instance, of the wild type. And so also we found that if we looked at another and we don't see this here. So basically, what occurs for the MDX, the standard mouse, also occurs for the this is an exon 52 null mouse. These are almost coincident, these lines. So if I look at this growth here, so this shows the coincidence of the 2 different MDX strains, wild type exon 52 null and exon 23 stop codon. So they're almost identical. What happens to volume? If we plot volume in the same way, that is the signal of phalloidin, then they follow very similar sort of overall line over this period, but they are nowhere near so extensive. So this is black. This is the reds are wild types. The blues are MDXs. And you can see that, again, the MDXs are bigger, they grow faster, but nowhere near so much. So they're only about 20% bigger at this stage, whereas they've got over twice as many nuclei. So the total density of myonuclei in MDX muscle shoots up, it doubles. And no one has really taken any account of this or tried to explain it in molecular terms. And we don't know whether it applies to other species either. So when you see two such coordinated patterns here, you might think there is a strong relationship between the number of nuclei and the size of the fibers. But we can look at that because we have data on individual fibers. So this is the wild type growth pattern. This is the actin content. This is the size of the fibers. We can ask whether these fibers here, which are the biggest ones, also have the most nuclei. And the answer is they sort of do, but not in a very coordinated looking way. And the same is true for the 8 weeks and also for the 14 weeks. So at all stages, there is a correlation. It's an overall correlation, but there's a huge variation between individual fibers as to the relationship between the number of nuclei they have and their size. And that also, I think, is under-investigated. We need to know quite why fibers differ from one another so much within the same muscle. So that's long-term dynamics. How do we measure short-term dynamics? Well, we can pulse label with bromodioxyuridine and see where the pulse-labeled cells get to and in what time. So this is the result of a 3-day pulse of bromodioxyuridine looking at a number of muscles, and we found that the triceps muscle showed we can count the number of fibers, the proportion of fibers that are marked with a nucleus, with a labeled nucleus, and in the triceps we got about 10% over 3 days, twice as much as the other two muscles we were looking at. And so we modified this protocol to look at what would be the result of 3 days labeling, 6 days labeling, and 9 days labeling. And we were expecting them to be additive, and indeed they sort of are. So this is the whole muscle here that's from a mouse that's been given 6 days of bromodioxyuridine. And this is just to show that it is labeling up the muscle fibers. These are the small muscle fibers that have just been labeled. And if you count the number of fibers, the proportion of the whole muscle that has labeled fibers, then you can see that there is a continuous increase between 3, 6, and 9 days of labeling in the MDX, which is not measured, not visible in the wild types, and it's much, much slower in the DBA2 MDX mouse, which is not the point of this talk. But so this slope here is about 2. So that means that 2% of all of the muscle in fibers in that muscle are being replaced by new fibers per day. And that's quite a lot. That's quite an extensive degree of turnover. So that's one way of measuring. There are other ways of measuring. So the other thing is we can give bromodioxyuridine for 24 hours, and we can look on the same day, at the end of the same day, the next day, the day, 2 days later, and 3 days later. And what you see is that even at the end of the first day, there are muscle fibers which have a central BRDU labeled nucleus. So this nucleus here was 24 hours ago in a satellite cell and was busily replicating its DNA. Since then, it has undergone at least one cell division, differentiated and fused into a muscle fiber. So this is a very rapid turnover. And it rather argues against the idea of counting satellite cells as a measure of myogenesis, because they're not satellite cells for very long. They spend a very brief interval being identifiable as satellite cells in so dynamic a process. So by 24 hours, almost all the fibers are labeled that you see by, and 48 hours and 72 hours, they have matured into muscle fibers. This is a very rapid process.
5 minutes left.
Pardon?
5 minutes left.
Okay, yes, I shan't be long. So the other thing we can do with bromodioxyuridine labeling is to injure the muscle at a known time with a myotoxin and subject them to single days of BRDU labeling and ask when these fibers, the cells that are responding to this injection, were actually in mitosis. If we look here, this is for 6-week-old mice, we can see that even the next day, there's a very high number of labeled muscle fibers, both in the wild type and the MDX. When we look at older mice, they are not labeled in the first day. So somehow these young mice have cells that are primed to instantly go into myogenesis, whether they're MDX or not. And that's not true of the older mice. So what is the problems for repairing muscle if you're a satellite cell? Well, satellite cells constitute about 2% of the total nuclei in a muscle fiber, and they've got over this sort of 3 to 4 days period at the most, usually 2 to 3, to provide enough committed cells to repair this muscle fiber. And to do that, they can only do that. It's a 50-fold increase. The only way that each satellite cell can provide, the average satellite cell can provide enough cells to repair this muscle fiber is by serial symmetrical division, a doubling on the number available on each division. The input from asymmetric divisions here is trivial, and you can model this to show it's so. So we know what the variables are that we can quickly build an instant model of that. So the picture we end up with is that when you are looking at regeneration here, it would be achieved by single satellite cells undergoing two asymmetric divisions one daughter going into the fiber that it's repairing, that it's growing, and the other carrying on to maintain the population. If you look at regeneration, then you've got a much bigger job on your hands. Each satellite cell has got to provide 50 cells that will repair the muscle. And so that can only be done by massive symmetrical division. And those nuclei go into the muscle fiber and repair it. The point I would like to make about this is that these are two totally different processes. How do these cells know when to stop? They don't touch one another on a fiber. The fiber is about 2 millimeters long at this stage, and there are only about 5 to 8 satellite cells on there. They won't see one another. They can't signal between satellite cells as to when they stop. It must come from the fiber, I think. Or either that or they're counting in some way. In this process with repair, you are building a very large number of cells in a group, and they can talk to one another. And I think there are a number of papers about suggesting that this notch signaling is very important in telling these cells when to stop. So these two processes are quite different. It's very likely that their signaling that's happening in these two processes is totally exclusive to that process and is unlikely to be compatible. It's unlikely that this process is going to be compatible with that process. So I think there's every evidence that part of the problem for Duchenne boys is that they are trying to do two incompatible things at the same time. So the people who did this work were James Novak in my lab in Washington, and Davi Marzala and Marie Nearing. And the whole thing was really started up by Bill Duddy's work on developing the measurement methods. So that's my talk. Thank you very much.
Thank you, Derek. That was really good. We have 1 minute for question.
We have 10 minutes for questions. Start, Gillian, please.
10 minutes for questions?
Yes, of course. To maintain the rules, you have to break the rules.
How Italian.
And I will start. Oh, Feliciano, are you talking, asking?
No, I'm present. I'm listening. I don't have a specific question yet, but maybe I will come up with one.
Well, I have my hand up.
Pete has one. Pete Summit has one.
Interesting, Terry. So in the regeneration paradigm, where you've got all these symmetrical divisions going on,
yeah,
when are you getting the self-renewal happening? I mean, obviously the satellite, do they maintain it all the time, or do they go through certain divisions and then?
Well, I'm as mystified by it as you are, but I mean, it is the mathematics is such that you cannot avoid that as a process. You can't get enough cells without that happening. we know that it's very short and episodic. Perhaps it's what happens between the episodes that keeps your satellite cells going. Also, the fact that you're probably only using a minority of them. So there's a paper from Tierney's group, from Alex Sacco's group, showing that during growth, you maintain a broad clonality, whereas during regeneration, there is a much narrower group of much bigger clones. And so that would fit entirely with my story, or our stories would fit together. And so I think we need to think of these things as not being, I suppose regeneration is an episodic event, and I think now our Biromidioxuridine labels show that each little lesion is dealt with as it occurs, whereas growth is a thing that's happening body-wide into all the muscle fibers at once. And so the questions are quite different. And the point I'd like to make really just is that they are probably not talking to one another, those two processes. In the mouse, it doesn't matter because the mouse largely does them in separate parts of its lifespan, whereas in boys, they're going on for 18 years.
And you're superimposing the two events at the same time and confusing the satellite cells.
Well, yeah, that would be a simple way of looking at it. Yeah.
I have a question too. Did you check if the macrophages are in the same numbers during early life and in adults? Because
no.
The roles of macrophages is not only to remove
yes, I know.
necrotic material, but if there is no necrotic material, there is.
Oh yes, well I mean I showed some pictures, and the cells that were labelled within the day in these lesions were all inflammatory cells lying between the fibers. They disappeared by 2 days out. So actually counting numbers of macrophages is, again, you're looking at a highly dynamic category. And so cells are in and out of those lesions very quickly, I suspect. And so counting them any one time only gives you a photograph of a moving dynamic picture.
Therefore, there are no data on the number and quality of macrophages.
I'm sure there are.
In the early phases of the life.
My wife,
because this is a major difference between.
Oh yeah, I don't know.
Young,
yes.
And old people.
Yeah, we did a paper on the delivery of morpholinos, which we think occurs in partly by macrophages. And so we have got counts of macrophages. We've looked at them over the 3-day period around say giving a morpholino antisense, and there are a lot there, and they fill up they pick up the morpholino, and we think hand it on to the myoblasts, but they're always there. But as I say, it's very difficult to pick up the dynamics of macrophages because they move so fast. They're in and out of the vasculature, I think there are now some recent papers on a resident and a immigrant population of macrophages, distinguishing between the two. And those are recent papers, and so I've not really had time to digest them properly.
Ask a question.
Yeah. Hello there.
Hi, Terry. I thought so. mTOR is dysregulated massively in Duchenne and also involved in the surveillance of stem cells, and so might be that mTOR pathway is involved in this phenomenon that we see.
Yeah, I don't know. I think almost no one has looked at the nuclearization of dystrophic muscles outside the mouse. So the mouse has been sitting around for 30 years. We were the first people to pick up the fact that there are twice as many nuclei there. What relation would that have to mTOR expression, for instance? You've got twice as many nuclei maintaining the same fiber.
Well, might be tested experimentally with mTOR inhibition.
I don't know. Yeah, we need to look outside the mouse. I think we need to look at human and other dystrophic muscle. We can't do that on single fibers. But we could do it stereologically. I'm trying to persuade people that stereology is an important thing to do.
Hi, Terence. I have a question.
Yeah.
Just a simple question. It's about, well, it's basically two questions. The first one is, have you seen or have you checked whether there's any difference in myonuclear accretion between slow type and fast type fibers?
No.
And the second one is, as you suggested, there are some satellite cells which should be primed or quicker in taking up muscle fibers during the juvenile stage.
Yeah.
Would you think that these are the myogenin positive satellite cells that have been suggested by Rantanin a few years ago?
Yes, I think so. I think they may be the ones with the containing RNA, 5 RNA. That would be a possible thing. The fiber thing is more difficult. We have totally failed to identify slow and fast fibers with antibodies from those isolated fiber populations. The background is too high. I've been talking to Pascal Mayer about the idea of using his sample to see what's going on. And several people who've been doing single nuclear genome work, gene expression work, have really not noticed or have not paid attention to the fact that half of the nuclei in a Duchenne multistrophic fiber are in the middle. They're the central ones of the big.
Yep.
The other half are still peripheral. And so, are they different? Are they different in what genes they're expressing? I think those are all questions that could be answered by people who are cleverer than me.
Thank you.